think how many people are out there? Like 32. Okay. So that's already too many for a Saturday right after the Sarah. So what I'll do is I will share my screen first, and I'll show you some of our works that got published in the last 5 years since I first set up my group back in Indian Statistical Institute, which is my alma mater. And after that, I moved to Delhi. And right after, I mean, just 6 months I spent in ISI Calcutta, where I did my PhD. So, and then, I built a wonderful group with a lot of PhD students from across different institutions. And today, I'll present their works, okay? And many of them have now gone for the postdoc in good places. So let me know if you can see my screen. Here I share. Okay, so is this visible to everybody? Vibhor, is this visible?
Yes.
Okay, all right. So my talk is about how to deal with dimensionality of single cells, right? Because single cells are sometimes enormous in size, and they are noisy as well. So the first problem is how to make fast algorithms that are scalable, and the second problem is how to ward off the noise in the single cell data. So first question is why single cells? So I think in the last 5 6 years, it has become very prominent in India. I know several groups now who work on single cells, and I am connected with at least 70% of them by some or the other way as collaborator. And why it became popular? Because single cells allow you to see the heterogeneity in the tissue. So if you see the left side photograph, it shows a tissue where if you look at it in the microscope, then you will see the heterogeneity. It is like a portrait, right? There are so many different colors and contours and texture, but all these things get diminished or blurred as you do bulk RNA sequencing, right? Because all your heterogeneous cell populations don't show up adequately in the averaged signal. Okay, so this is why single cell genomics has now become, I would say, synonymous to genomics. So it's a big statement I'm making, but it is no more a fancy tool. It is a very standard tool these days, because your next generation sequencing revolution started in 2009, right? 2008, 2009. And if you look at the first single cell paper, it came out, I mean, a couple of years after that only, 11 to 2011 or 2012. So I don't take any credit of doing something extraordinarily new. This is a very cliched field now, but still the problems persist. Now, what is happening is the single cell data is growing in volume, right? And the platforms are becoming cheaper and cheaper. Now, the question is, how do you manage to understand this deluge of data and still get a lot of good insights about the biological system? So my training is into algorithms. I self-taught myself biology. So I'm a half biologist and quarter to one kind of a engineer. So therefore, you may, if you ask very deep biological questions, I'll struggle a bit, but you are free to ask me. Okay. All right. So single cells are indeed a new prism for molecular biology. So if you look at the bulk RNA-Seq data, you see an average expression level of the tissue. Whereas if you look at the single cell RNA-Seq data, you can actually characterize the different cellular subclones or the subpopulations. You can think of cancer. Cancer is extremely heterogeneous, right? Every cancer is different from every other cancer. And also it evolves with time, right? So just imagine what could be the role of single cells, because you can find out the cancer stem cells, which become resistant to the drug, right? And thereby you can treat it specifically for a particular person, right? So you can implement personalized medicine approaches based on single cell profiling very easily. We are doing a lot of those works, but unfortunately, I didn't get the time to put all these things together. But the massive stress point for my lab is now how single cell can be brought from biology, basic biology, to translational biology. Okay, so but that could be, I can talk about that on a different instance, not today. Now, what all we get out of single cell sequencing, unraveling tissue heterogeneity, right? You can study each and every cell type. Unknown cellular states can be unraveled, right? Because a lot of times the cells that you see and you think that they are of different types, they are actually at different states because you are actually taking a temporal snapshot, right, at one particular point in time. And therefore, it doesn't talk about the past and the future necessarily, although recent works suggest that you can actually derive the past and the future of a cell from the single cell expression. And those are very exciting directions of the research in single cell genomics area. And disentangling differentiation processes, right? As a byproduct of what I said, if you can know the future of the cell somehow, then you can actually, chart out the evolution process. For example, how a tumor evolves, how new cell types get generated, how they become drug resistant, or how a simple organism develops, right, from embryo phases to more mature phases, right? And of course, discovery of rare cell types. That is a very pertinent question in many, many fundamental biological areas, right? So we'll look at some of these use cases in the following slides. So this is just a very iconic picture of how you can unravel the evolution of cells. How can you draw the cellular evolution, developmental trajectory of the that is explained by a bunch of single cells? And those are very extraordinary kinds of works which I'm not discussing here, but you can check out later on the work by Peter Karchenko's group in Harvard, right? So look at the basic paradigm of single cells and bulk RNA sequencing from a more methodological point of view, right? So you have matrix of genes and few samples in case of bulk RNA-seq. In bulk RNA-seq, you typically create 2 to 3 replicates of the tissue, right? Whereas in a single cell, even on a single subject, you get thousands to millions of cells profiled at a very low cost per cell, right? So, and the matrix that you can see on the right hand side is slightly more choppier. That means your sequencing depth cannot be that high. There are a lot of different kinds of errors also, and cell to cell variability as well that take place, and that confound the overall sequencing output. So the challenges are following: large dimensions, high levels of noise, and lack of methods that scale. Now, a typical single cell analysis pipeline looks something like this for ones who are not familiar. Raw matrix is given, which is your gene expression matrix, where you have the count of each and every molecule across the cells. And then you do gene and cell filtering, you do normalization, you do dimension reduction just to visualize how your cells look like, how are the dissimilarities and the similarities. You can also perform rare cell discovery. This is also an unsupervised task. And then you can perform clustering and differential expression analysis to characterize each and every cluster and what are the typical upregulated and downregulated genes. Finally, you can perform go term analysis, right, to understand the functionality imparted by a bunch of upregulated or downregulated genes. There is another very exciting direction, or I would say kind of an insight that you can derive from single cell data, which is serotype analysis, right? So you take any system under development, right? It could be any particular organ, it could be a diseased tissue or anything, right? And you can actually do single cell profiling and you can infer how cells come from each other. What is the trajectory of cellular evolution? Right? Those kind of inferences you can make very easily and in a quite robust way. So this is the overall picture of how the basic analysis of single cell data is carried out. So we, back in 2016, introduced a revolutionary concept in single cell genomics, which is now followed by probably every leading lab in the world, which is basically assigning a cell a zip code, right? Just like your house address would typically have a zip code, we thought that it is more apt since single cell data is voluminous, very huge, right? Uh, it- we can save a lot of time of analysis simply by hashing the cells. And hashing means assigning a binary vector or a bit code to a cell. And that actually helped us enormously improve the speed and the quality of the analysis. So a lot of works that we did subsequently in 2017, 18, and 19 were based on this hash code of the single cells. As I said, even if you pick up a lab from MIT, I don't name the lab, but they follow this protocol, right? So now it has become kind of very standard way to represent a cell because it becomes computationally lighter. Now, if I just try to explain you what hashing really means, so if you see here, locality sensitive hashing is just one family of hashing, right? There are many other ways of assigning the binary code to a particular data point. In this case, which is a cell, there are many different ways, but locality sensitive hashing is the most primordial way to do that. And I must tell you that the inventor of locality sensitive hashing is an Indian origin guy. His name is Rajiv Motwani, who was in fact the mentor of the Google founders, right? So he is a very, very celebrated Indian computer scientist, if not the most celebrated. Okay, I think he's the most celebrated Indian computer scientist whose entire career was spun in US in Stanford University. He was a professor for a very long time. And one of his other celebrated algorithms is PageRank algorithm, which many of you who are from computer science would know. So what is being done here is that you have two balls, right? You have, and you can think of these balls as single cells. You have a, yellow ball and you have a red ball, and you have two-dimensional space. So you can imagine that this x-axis could be your one gene and the y-axis could be another gene. You can also ask me why only two genes, because we have 20,000 different genes or more than that. You can actually, it is hard to draw. It is actually hard to draw all those dimensions in a page, and therefore we are just sticking to just two dimensions here. So what is happening here is we are drawing some random hyperplanes, right, of the same dimension of the data. And these hyperplanes actually can run, there are different ways of drawing these hyperplanes, okay? And these are quite mathematically involved, theories which one has to understand. And I really don't want to derail and get into all those things. But what happens is if you just draw, let's say, five hyperplanes, and you simply look at how these cells are positioned across a particular hyperplane. For example, if it's in the left hand side, you can assign it a 0, otherwise you can assign it a 1, right? So in this way, for these two cells, I have come up with a binary bit code. And this binary bit code actually expression values of the cell, and you converted that into an extremely low dimensional hash code or a binary bit code. And that has a lot of very good qualities, right? It preserves the cosine distance of the cells. And it is very powerful. It is very easy to handle. It's a bunch of matrix operations that you can perform even in a graphical processing unit, and you can speed up your analysis substantially. Now you can measure the Hamming distance of these bit codes. Like if I want to understand that how similar or dissimilar these cells are, you can simply measure the Hamming distance of these bit codes. And as you can see, the only place where they're different is the last one. And therefore you have put a cross here. and you can approximate the distance as having distance divided by the signature length. The signature length is 6, which in other terms means that you only have 6 hyperplanes. So 1 upon 6 becomes your approximation of their cosine distance, which you can see here through this expression of cosine of h by b times pi is approximating your original cosine distance between the cells. So that is the crux. And then we applied this in many different problems of single cell analysis. I'll not spend a lot of time on that because these are extremely specialized. And I appreciate that not all of you come from that background. I'll just tell you the broad results. The methods are too difficult to understand just in this 40 minutes talk. So the steps of the drop clustering pipeline, by the way, you might know what clustering is. Clustering is basically grouping the samples of the cells, right? Because single cell analysis, by its virtue, unsupervised problem, right? Somebody gives you a bunch of cells from your PBMC, from your blood, and you are asked to cluster them to understand what are the different cell types or whether there is a new cell type. The clustering means grouping of the cells. And clustering is an NP hard problem, right? Finding out the most optimal cluster based on certain distances is a computationally intractable problem. Although it sounds funny these days because we do clustering as a normal standard process for analysis of any data. Pretty much everybody understands what clustering is. So we actually tweaked the existing pipeline substantially. We published 2 back-to-back papers in nucleic acids research and bioinformatics to describe the drop cluster pipeline. So it involves pre-processing of the data. That means starting from the gene expression count data, you get the batch effect removed version of the expression matrix. And I would really insist you to check on our bioinformatics paper on this idea of using ranks to remove batch effects, right? So we, for the first time, built an algorithm where you don't use the values of the gene expression, but we use ranks in a very clever way to get rid of the batch effects. And batch effect means even the same cell line or same set of cells may appear differently if they're processed on different times in different labs with different chemistries, right? And I'm sure some of the previous talks might have highlighted some of the notions of batch effect, so I'd not expand on this anyway. So then we implement something called structure preserving sampling, right? What is structure preserving sampling? Because if you do a random sampling, you can imagine that a bunch of cells that are coming from your blood, the T cells will be dominating, the dendritic cells will be probably very, very rare, right? So if you just blindly sample your, just to make the problem easier computationally, let's say somebody gave you 1 million brain cells, right? And now clustering of 1 million brain cells is extremely difficult. So you want to subsample that, but subsampling randomly would potentially wipe out a lot of rare cell types. And this is why we implemented a technique called structure preserving sampling, where you preserve the structures in the data. That means the clusters of the structures, right? So we kind of did a pseudo clustering and then did a cluster wise relative subsampling. Like the cells which are large in number, we took less relatively as compared to the cells which are small in number. Then we also tweaked the principal component analysis, how we select the principal components, so that we changed to get better representative genes for the data. And we performed hierarchical clustering as standard, and we did post hoc assignment of the cells which fell out of this subsampling. Now, this is a result showing different cell lines. Now, if you just follow the legends here, you see the batches are in line 1 and 2, the squares and the circles, and then these are the different cell lines. And they are processed in different batches. So you can see, although this is hard to see here, but you can see certain batch wise in the raw data, right? Like the cells are falling out, like coming from one batch is tending to go together as compared to because ideally what you want is you want the same types of cells irrespective of their batches clustered together. So this is how dropclust performed on this data, and as compared to dropclust, the other methods kind of didn't do very well. Scanorama is a Nature Biotechnology paper by a group in MIT. They sort of used a similar kind of an idea of ours, and then they sort of got a good result, I would say, in this data. Now, this is how the structure preserving sampling helps. So as you can see, what happens if you do sampling randomly? So the bottom one is the random sampling. This is a 68,000 PBMC data, blood cells. And I am just plotting like 500,000, 1,500, 2,000 cells selected randomly. And as you can see, with structure preserving sampling, you are tending to keep more cells from clusters which are rarer, right? So you take more cells from, for example, dendritic cells. So here one example has been shown. This is megakaryocytes. Now you can imagine that in this figure, megakaryocytes, you just have one cell. Dendritic cells, we have so few cells. So we are actually preserving cells coming from small clusters very carefully, and thereby we are getting a better representation of the entire population without compromising on the consideration of the minority cell groups. We also identified a major problem with the principal component analysis, the way it is done today, because principal component analysis for example, in this artificial data, the direction of maximum variability is this direction, which is marked as PC1. But just imagine if you take a dot product, you project all these cells, the green cells and the red cells, on the PC1 axis, you will not be able to distinguish between which cells are coming from which lumps, right? So it will probably look something like this. Now there are two populations, but after projection on the principal component one, you see they're almost inseparable, right? You don't see a separation. You don't see separate modes here. Whereas if you look at the principal component two, then you can see the modes are appearing clearly. And just based on this simple observation, we decided to choose principal components not based on the variation that it explains, which typically everybody else does, but we chose the principal components based on the number of modes that it represents after projection of the data. I hope it is clear. So here is an example on the real data. So what we show on the top figure is that the total variation explained by each and every principal component. So the light blue is the variation, right? So as you can see, the number of modes is not necessarily totally correlated with the amount of variance each principal component explains. So we just took those principal components which show high number of modes. So here is what it looks like. So if I project all the PBMC cells in this data on the top principal components according to the variance, and the top principal components according to the modes, then how different they look like. So you can check it out here. Like the best component is 2. You see the number of modes on the 2, component 2 is highest. And you see the different annotated cell types are pretty much spaced out well, at least much better than principal component 1. So this is the best principal component based on the variation. This is the best principal component based on the number of modes. If you just follow these two figures, you'll see that all these cell types are actually spaced out very neatly, much more neatly, I would say, as compared to this particular thing where all these things are tending to clump up together, right? And similarly, you can see the following principal components. So we have some decent results which were computed in terms of adjusted RAND index, and the time consumption was good, right? I mean, is quite acceptable. In case of Dropplust, you see minimum time taken as compared to the other methods. And the memory requirement was also reduced significantly in our recent most version of the Dropplust. It is available freely for your use. This is another result, and you can see how neat all these changes that you have done in the pipeline give us a much more cleaner segregation of the different known subtypes of the blood cells as compared to the then popular methods like ScanPy or Ranger and Surat, right, which are still kind of, Surat is still the industry standard, I'd say. So this is one of the publications. I forgot to put the other one in the bioinformatics, but this happened in collaboration with Sangamitra, who is the director at ISI and happens to be my supervisor, and Himanshu, who is a leading immunologist and an INSA fellow last year, and he is in ISAR Bhopal. Okay, so he helped us in analysis of the interpretation of our findings with the immune cells. Cell Atlas Search, I guess Bibhor has already talked about. So that's the collaborative work with Bibhor's lab in IIIT. And it is quite a remarkable achievement, I would say, because we sort of created the first ever search engine for single cells. So it came right after the Dropclust paper, and it uses the same principle of hashing, right? And however, it also infuses the principles of GPU computing. So this is hashing plus GPU computing is kind of a cool combo that makes search of large volumes of expression like expression profile. So similar expression profile search possible, right, online. And that is still available online. And I think Vibhor's lab is working on extending this search engine on other omic dimensions. And this is how it works. I just don't want to spend too much of time. It basically hashes the expression profiles into different buckets, which makes search of a particular given expression profile by the user easy. It lands the user expression profile into one of these buckets, which is a hash code basically, and thereby you can very quickly determine which are the similar cells or the bulk RNA-Seq samples. This is how the output page looks like, where you can also see projection of the single cells and the bulk RNA sequencing data and all these cool features. I think this is also getting quite a bit of traction, and it was kind of an ahead of time project, I would say. It came even before the single cell atlas initiative took place. I'd skip some of this works. So this publication you can check out on Nucleic Acids website. A major contribution of our group is to give an algorithm that really scales on really large data set. And let me remind you that one important reason why people do single cell sequencing is to identify unknown cell types or cell states, right? And we realized that back in 2017 when we started working on this problem. Actually, we started working on this problem in 2016, right after I established my lab. And we realized that the existing methods for the single cell search, rare cell discovery, were very slow and very traditional. Okay, so they use statistical ways and clustering and whatnot for finding rare cells. I found those methods to be quite arbitrary. And at that time, there was a boom of DropSeq, right, which allows you to sequence millions of cells or at least tens of thousands of cells effortlessly, I would say. And we asked that how can we enable rare cell finding on such voluminous data? And that was the genesis of FIRE, right? And you can search any kind of rare cells. For example, there are even dendritic cells have 4 different subtypes or 6 different subtypes that was described in a science paper in 2017, I guess. So they are apparently, unfindable, I would say. You cannot identify rare cells very easily given a large data. So the question is, how do you do that? And we developed a very monolithic algorithm, I would say. Monolithic means this algorithm is very basic, right? It is not complicated. It does not have too many branches and wings. It is a very simple thought. So there is a, again, it is based on hashing, right? So here is a schematic diagram that describes how this algorithm works. You have these cells here, which are high dimensional vectors you can think of, right? Because each cell is 20,000 genes expression. And now you have some of these cell types are actually quite rare in number, right? You see the pink cells are too many, the yellow cells are too many, whereas the black cells are too few, right? So what we said was we can hash these cells, right? Again, we can create the binary bit codes, and we can put all these cells into one cubicle, right? Each hash code is a cubicle. You should imagine that a hash code will be shared by many different cells, because this is kind of a crude zip code. Like in a single zip code, too many families reside, right? Too many people will have the same zip code in the same way, but they will be co-localized geographically, isn't it? Like we all stay at different places. If you look at the coordinates, right, the longitude and latitude. Whereas if you look at the zip code, then we share zip code. For example, all of us staying in triple IT share 110020, right? In the same way, so it is kind of a lower resolution. So hashing is a lower resolution, and that gives you a lot of cells or small number of cells across the cubicles. Some of the cubicles can actually be blank. It is absolutely okay because you are creating these zip codes randomly. Okay. Now we did this exercise multiple number of times, right? It is kind of a bagging or boosting you can think of. So it is a bagging of hash codes in a way. So and we estimated the probability based on how many cells share the same cubicle, okay, divided by the total number of cells. How many cells are there in this cubicle divided by the total number of cells is the probability of how rare the cell is, right? And it is quite intuitive. Now we took these probabilities and we used Fisher's method to combine this empirically calculated probability values using this formula here. As such, what happens? For the rare cells, we get very high values, combined values, and for the non-rare or abundant cell types, we get smaller values. Well, this slide is misplaced. Yeah, here is an example which is easy for you to understand, like the same PBMC data of 68,000 cells. And I am just pointing out the 0.25% of the cells, depending on the high fire score. The top 0.25% of the cells in terms of the fire score. Now see which cells are lit up here. It is interesting to see that megakaryocytes are lit up here, right? So 0.25%, you only capture megakaryocytes, and we all know that megakaryocytes are extremely, extremely rare in the blood pool, right? And then as you increase the number of cells to 2%, you get to see dendritic cells here. And if you increase it more, you get to see certain types of the monocytes as well. We benchmarked it on simulated data set. It is useless to talk about this. This will take a lot of time, but here we knew it is kind of a controlled experiment where we created the data using the existing data only, where we basically took too many cells from one category and too few cells from another category, and that we declared as the rare category. And then we sort of annotated them as rare and not rare, right? And then we tried to see that in our way of identifying, calling the rare cells, how accurate we are. So here we present some of the statistics. We sort of computed the F1 score as well. That looked pretty favorable in our case. And needless to say, it is very, very fast, right? If you give me 1 million cells and a reasonably good workstation, it will not take more than 1.5 minutes. If you give me 40,000 cells, it will take 10 seconds or 5 seconds. So it is kind of unbeatable, right? And we use this to find out rare cells from a publicly available mouse brain data. And we sort of found, coincidentally, we found in situ images of the respective mouse brain area in the Allen Brain Atlas, which is a Microsoft initiative. You should check out. It's a beautiful website where they preserve. These days, they also have single cell data. They have bulk RNA-Seq data of the different brain regions of mice. They have the in situ images. You can actually rotate the brain and see each and every plane, the coronal, sagittal planes of the mouse brain, and you can really see whether the markers that you have somehow found out, like I found out these three different cell types here, which are pretty interesting. And I sort of tried to see if they have some markers which are special just to them. For example, you see this R6, you know, has a lot of markers which are differentially expressed, and the same is true for this and all, right? So we could identify some of these markers expression in the in situ images, okay, and that indeed indicated a small rare population. Only upon zooming the picture we could see the traces of this particular marker. Now this is not our data, we just used the data from the Allen Brain Atlas. We could also identify 6 different subtypes of um, dendritic cells right from the PBMC data without any sorting. By the way, as I told you, in 2015 or 2016, I guess, there was a paper in Science FACS sorting dendritic cells, because dendritic cells are extremely rare. If you want to study the populations of the dendritic cells, you need to FACS sort them. And then you can create multiple single cell profiles of that. But we asked that can we discover the same set of cell types as from a simple PBMC data? Then it turned out that out of 6 cell types described by the authors of the science paper, we could actually identify the 4 subtypes of the dendritic cells, which itself is a very, very small population, quite effortlessly. And we could also identify the genes which they identified or they claimed to be differential across all this population. So this is kind of a good validation, an independent validation of our study. Yes, this is the paper that I was talking about, which talked about the heterogeneity of dendritic cells. This is our publication, and this happened in collaboration with Jaydeva, who is the current head of electrical engineering department in IIT Delhi and a very good collaborator. And Ashi Prashant were the lead authors of the paper. Now, this is my final thing, and I'll stop after that. Okay, so we are also partly interested in how many more minutes I have. Like, I know it is already the time, right? Because it was a bit late on, so I'll take probably 5 more minutes and finish. So there is a fundamental dichotomy in the space of statistics, right? Because all of you know that finding differential genes is a fundamental problem of biological research between two groups which genes are, differentially upregulated or downregulated. So single cell expression is, so this is how a differential expression plot looks like, the heat map looks like. So this gene is upregulated in this cluster and so on and so forth. You see this kind of a staircase representation. Now, you should remember that single cell expression distribution is a lot more wiggly as compared to the bulk. You see different bulk replicates like myoblasts differentiating myoblast as 0th and 24th hour. You can see pretty much overlapping, indistinguishable distribution, whereas in single cells it becomes really, really wiggly. And also the number of expressed gene counts is also very different across the cells, whereas for the bulk samples, they are very stable. So you can imagine the amount of noise you have in the single cell data. So we asked if we can create a differential expression method that does not really rely on the expression values themselves, but ranks. But you know, the problem of ranks is that ranks are robust, but the problem is you cannot do a parametric modeling of rank. So we took on this challenge head on, I would say, and we said that, look, can we create a parametric model for a rank-based test? Because a parametric model gives you statistical power, right? And rank gives you robustness. So can you merge this two? And I stumbled across this wonderful paper in PLOS One. This is probably the only paper in PLOS One that I have ever read. So they said there is something called rank ordering distributions, where the ranks and the frequencies, right, are very nicely related by this equation. This is a generalized beta distribution sort of a thing. Now you can see this picture. This is an abstract art. So they have measured the rank of the rectangles and the area of the rectangles, and you see they follow this beautiful pattern. This is something similar to the normal distribution, I would say. Okay, that is seen every time. So it is there in fine arts, it is there everywhere. They have shown multiple phenomena. And if you are interested in this equation, this A is a normalizing factor, N is sample size, R is the rank, Y is the frequency, A and B are the parameters as in the beta distribution. You guys would know. We showed that you can actually fit beta distribution very nicely on single cell data. And we further showed that you can actually develop a statistical world test to sort of compare these two populations. So you can actually compute the log likelihood estimation equation to log likelihood estimation of A and B, because these are the two parameters you want to estimate. And you can compare this to the set of parameters is using world tests. So this is a world t-test statistic. And for that, you need to compute to very complicated things, like the variances are complicated to compute. So variances come from the Fisher information matrix. So these are very standard mathematics, right? There's nothing very peculiar here. So this information matrix is basically how the log likelihood changes with respect to the parameter values A and B, right? So there are second order partial derivatives of L against A and B, right? The log likelihood against A and B. Well, so the performance, as you can see on our own data, which comprises, I would say, one cancer cell line and fibroblasts, we could see with respect to the known genes to be differentiated up and down regulated. We could see that we are the best in correctly calling them. And this is just the top 10 genes across the two groups. And it is also very robust against dropouts. I don't want to spend time on that. You can check out the original paper, which is this. This got published early this year in Genome Research. And this was done in collaboration. The main collaborator was Abhik Ghosh. Abhik Ghosh is an assistant professor in Indian Statistical Institute. So he's a statistician, quite a prolific young statistician, I would say. And of course, there are other people. Naveen provided the data. He is the senior director of research in Fluidime, US. And Krishan was the first author who is the first PhD student from my lab, and currently he is in Harvard Medical School as a postdoc. So the conclusions are simple. Hashing is kind of a zip code that helps speed up and bettering the quality of the analysis. Ranks are more robust, but you need to be statistically solid or mathematically appropriate to use ranks. You cannot just say, I take ranks and then cluster, it will not work. Okay, it is not. So you really need to spend time in devising statistics and mathematical arguments along that direction. And of course, low dimensional zip codes make the cells in the bulk profile searchable. And this is a set of collaborators that I'm actively engaged with currently. I got funding from a lot of different sources, which are shown here. And this is my group, part of my group, not the entire group. So Krishan is the RNA-Seq guy, and Smriti, Chitrita, Namrata, and Sarita. So they're pretty much all of them will finish their PhD in the next few months. So with that, thanks a lot. I'm open to questions if you have any.